Hi, everybody. Welcome back. In this mini lecture, we're going to really begin to dig into really the heart of the class with a discussion of one of the grand challenge problems in cellular physiology that is going to underlie and motivate a lot of the work in the first half of the class. That is the concept of cell fate decisions. So let me switch over to the view of the slides and get us started here. So this first portion of the course is focused on cellular physiology. Why have we chosen to start with this particular aspect? Well, part of it is that the cell itself is really the fundamental unit from which all branches of the tree of life derive. This is a picture. I actually have this on the wall in my home. Schematic of the relationships between all existing types of life. And I'm sure that you've seen something along these lines in the past, representing the history of life, the history of evolution in unicellular organisms, plants, insects, vertebrate animals up here, and showing how they're all interconnected. But what you might not really think about, and what I think is one of the most fascinating things about this view, is this doesn't just show relationships, but it actually also shows, in some sense, an uninterrupted flow of cellular material. In order to get up here to a whale, that whale itself came from a single cell that derived from its parent, and that organism came from a single cell that derived from its parent. And as you go back generations and generations, you pass through evolutionary time down into the very early depths of the history of life, when all organisms are just single cellular organisms, and at some point down to when all of us, us being not just people, but all animals, all plants, all life on Earth share a single common cellular ancestor. Right? And that continuity of cellular material, of cellular processes is amazing. The cell is the fundamental unit of life. Certainly, multicellular organisms can exhibit behaviors that go way beyond the capabilities, the behaviors observed in unicellular organisms. But these higher order, if you will, these emergent behaviors of a large multicellular organism like a person or like a tree, these behaviors must in some way derive ultimately from how individual cells are acting and how they're coordinating their behavior. We can't understand the behavior of organisms in any really deep way. We can't understand organismal physiology. We can't understand organ or tissue physiology if we don't understand the underlying cellular biology. And so this is why I've chosen to start with this cellular physiology framework for this class. Now, cellular physiology can be broken down into various types of processes. It's not necessary to really memorize all of these terms, but it's good to become familiar with them, learn to recognize them at least in context. We discuss catabolism Together, catabolism and anabolism make up generally what we call metabolism, the processes that build up and break down molecules and convert them to energy. Catabolism is specifically the processes that break down larger molecules into smaller ones, either in order to produce energy, one of the most common catabolic processes is that of respiration, where we generate energy by essentially burning sugar. But also the processes that break down molecules into building blocks from which other molecules can be made are also part of the catabolic pathways. Anabolism are the processes that build up the molecules of life from smaller components, in almost all cases using energy in the process. These can be the processes by which energy is stored in starch, in glycogen, in fats. It can also be, it also includes the processes that make proteins, or that make the building blocks of these, that make amino acids, that make nucleotides, and so on. In addition to these processes that fundamentally involve modifying molecules, there are transport processes that move material either in and out of a cell or from one part of a cell to another. There are signal transduction pathways that really, in a fundamental sense, transmit information from one part of a cell to another, that somehow send a signal, for example, from the outside of a cell, relaying information, for example, about the presence or absence of a certain type of molecule, or a temperature, or a pH, and transmit that information into the inside of the cell, very often into the nucleus, where the next general processes come into play, though that is the processes of gene regulation. These are the processes that control which genes, we'll discuss a little bit exactly what this means a little bit later, which genes in a cell are expressed, that is, turned on, and which ones are turned off, and if they're turned on, to what degree. These fundamental processes, if you will, these low-level physiological processes that often can be described as a simple either chemical reaction or single transport phenomenon, these build up into cellular level behaviors. Cell movement. Does a cell move or does it not? If it does, how fast does it move? In what direction? The motion of a cell is driven by these lower level processes, some combination of signal transduction, sensing the environment, catabolism, generating energy that is able to create motion, and perhaps other phenomenon as well. Cell growth. Does a cell grow over time? Or does it maintain a constant size? In some circumstances, for single cellular organisms, for example, it makes sense for cells to grow and grow at least whenever energy is available. In multicellular organisms, it is very important that cells that have reached a certain size, the correct size for their function, don't continue to grow beyond that size. Cell division. When does a cell decide to divide into two daughter cells? Or when does it decide not to remain sort of a single cell? That decision process also has to be driven at some level by these lower level physiological or even biochemical processes. One of the perhaps most interesting um, cellular behaviors, and one that will, in the next sort of couple of slides, explore a little bit more about the consequences of this, is that of cell differentiation, um, particularly in multicellular organisms, but also in even in simple bacteria. Cells can exist in different states, different types. This is obvious again in multicellular organisms. We have skin cells, we have muscle cells, we have bone cells, we have nerve cells, which all are very, very obviously different types of cells. What drives a cell to remain its constant type? What drives cells to transform into a different type? How is that process controlled? At some level, it's controlled by these lower level physiological processes: signal transduction, gene regulation, coupled with the supporting processes of anabolism, catabolism, and transport. These are kind of a summary of the sorts of things that you might observe a cell to do. Once we move beyond cells into either multicellular organisms or into populations of unicellular organisms, we move into a whole other class of biology, but which again is driven by these more fundamental cellular processes, these so-called cell fate decisions. What do individual cells do? How do those individual cells behave? These cell fate decisions underlie what I think is one of the most fascinating problems in biology, the development of multicellular organisms from a single cell. Every one of us began life as a single fertilized egg cell that more or less looked like this, a sphere. And over time, this cell divided into a little cluster of cells. But then over many, many divisions, shapes started to form, structures started to appear, cells started to differentiate. And ultimately, you end up with something that, in my tremendously good art, looks something like this: a fully structured person with hundreds of different cell types, all in the right place, interacting in a fascinating way that not only drives our ability to live, but drives our ability to be in this class thinking about these problems. All of this came from a single cell through a series of cell fate decisions, decisions to grow, decisions to divide, decisions to differentiate, and decisions not to grow, decisions not to divide. In some cases, decisions even to undergo what is known as apoptosis, or programmed cell death. In multicellular organisms, for the sake of the organism, sometimes the correct decision, the necessary decision for an individual cell is that it no longer needs to really exist as a cell. This process of development of organisms, the field of developmental biology, again, is this tremendously rich, fascinating, and hard problem. But there's examples of these processes in many other contexts as well. In a healthy adult, your blood cells are constantly being replenished from so-called stem cells that rest in the bone marrow, And these cells go through a differentiation process, some of them becoming red blood cells, some of them becoming platelets, some of them becoming all of the various types of white blood cells. This cell fate decision processes happening over and over again. Every time a wound heals, if you get a little scratch on your arm and that heals, there's a whole process of cells moving into form into that area as part of the immune response. Cell growth being stimulated, where there was much slower growth previously. Cellular differentiation to make sure that the right kind of tissue is in the right place. Right? These are all things that happen in healthy individuals, things that we want to be happening in some sense. Things can also go wrong with these processes. The development of tumors, both benign tumors that don't really cause any deeper issues, and malignant tumors or cancers fundamentally derive from these cell fate decisions going wrong. That growth becomes improperly controlled. Cell division becomes improperly controlled. Cell movement can become improperly controlled. And cellular differentiation can be improperly controlled. In addition to these examples from human biology, and which can be extended to most vertebrate biology, the same sorts of problems apply even, and cell fate decisions are ongoing even for unicellular organisms. Many microbes can exist in both virulent states, where in the context of infecting an individual, they cause a lot of problems, and non-virulent states. Every so often we hear about an outbreak of some deadly E. coli that might have contaminated a batch of, say, spinach in the grocery stores. And hear about it. There's been a number of cases where a number of people have died from these infections. And this makes it seem as if E. coli is this nasty, nasty bacteria. But the reality is that E. coli is in every single one of us in massive quantities. It is one of the most common bacteria in the mammalian gut. And in the vast majority of cases, it does nothing harmful, and in fact is essential to healthy gut health. This normal state for E. coli is a non-virulent state. What is it about these other strains? These other strains have undergone some sort of a switch into a virulent state. You might think that it's just a mutation, and maybe these are just a mutant form of E. coli that happens to be virulent. And there are mutations that sort of can be involved in these processes, but it's a deeper issue than that. That essentially this ability to switch from a virulent to a non-virulent state is something that in some sense is encoded in the bacteria. It's something that the bacteria has a capability of, and some mutations make that transition easier to happen than others. Another very fascinating behavior at the microbial level is the formation of biofilms. Many single cellular organisms have the ability to exist in two different states again. One that is free floating, if you will, where the cell behaves truly as a single cell, and all of the decision processes seem to be made in a single location. There's no coordination between that cell and others. But many unicellular organisms have a capability of also existing in a form called a biofilm. A biofilm is a dense layered structure, often of many different microbes living together. And in biofilms, these microbes communicate. They send out signals into the environment, and they coordinate their behaviors in many ways. This transition from free living to a biofilm state is another cell fate decision. How does this sort of thing happen? Even when we go down to really not living things per se, or not free living organisms as we think about them. Viruses have an ability to, at least when they have infected a host, drive cell fate decisions that can affect whether the virus actively mounts an infection, if you will, and tries to reproduce rapidly, or whether the virus sort of enters a more quiescent, a latent state where it, in some sense, hides inside the genome of an infected cell. Essentially, at every level in the tree of life, cell fate decisions play a role, at least in some of the biology of these organisms. So because of this centrality of cell fate decisions, this is going to be one of the recurring themes that we come back to in the class. If we could robustly understand how cells of diverse types make specific decisions, there is so much that would be opened up. And we are beginning to get there, but there are still many, many questions that are still to be answered in this area. There's questions of fundamental scientific understanding, really the what, why, how of the problem. Under what conditions will a given cell make a particular cell fate decision? Why does that happen? What drives that process? Can we understand that decision process, that kind of signal processing issue, if you will, in detail? If we could do that, if we had that level of understanding of what makes cell fate decisions occur, we might be able to use this to make robust predictions about what might happen under certain conditions. This can be used for predictive biology, predictive medicine, understanding really what drives and under what conditions certain decisions are made would give us tremendous better ability to predict outcomes, for example, in cancers, both generally and under various treatment regimes, and could dramatically benefit treatment outcomes in that area. From prediction, we can also imagine going to manipulation. If we understand enough about what drives these cell fate decisions, such that we can make predictions about what a particular cell might do under particular conditions, then we might also be able to design manipulations that would make a specific cell make the decision we would like it to. Certainly, there are applications for that in medicine. That's really what we are trying to do when we attempt to design anti-cancer drugs. One of the things that we're trying to do is to dictate to cancerous cells to make different decisions, either to stop unregulated growth or to undergo apoptosis, to undergo other sorts of changes that would reduce tumor size or at least halts tumor growth in a robust manner. There's also a lot of potential applications in bioengineering if we have the ability to engineer cells to do specific things under specific conditions. So in summary for this first mini lecture, or second mini lecture, first on real content, there are a set of low-level physiological processes, fundamentally processes of biochemistry, that underlie observable cellular behaviors. These are the processes of metabolism, breaking down and building up molecules, of transport, of signal transduction, and gene regulation. These low-level physiological processes underlie cellular behaviors, including behaviors of cell fate decisions, choices to move or not, to grow or not, divide or not, differentiate or not. These are particularly interesting behaviors because they underlie a rich array of biology. If we could understand these problems in a deep and predictive way, we would have profound effects on biology and medicine. And in order to do that, it is almost essential that we are going to be able to build quantitative, mathematical-based physics and chemistry-derived models of these sorts of problems. These are complicated pathways. We may be able to get a qualitative understanding of how these systems work without mathematical and computational modeling, but there is no way we will have quantitative predictive abilities if we don't have a modeling framework to go with it. In the next several weeks, we will be discussing how we develop that modeling framework under different contexts and exploring how to use it to look at these kinds of problems. Right. With that, this mini lecture is over. There will be, in the next lecture, we are going to go through some of the underlying processes in a little bit more detail, discuss the mechanistic underpinnings of these basic biochemical processes, and from there we'll develop our ideas of how to translate these ideas into math and ultimately into computational models that we're able to explore. Okay. Take care and be well.